Okay, so hello everyone. Today is the session for dedication on session is dedicated to discussion on this third session we are going to conduct today, and that is on anti-neoplastic agents, anti-cancer agents. So in the last two sessions, we have discussed what is called cancer, what are the types of anti-cancer agents, and we started their classification. And among this classification, we have seen anti neoplastic agents in first class, that is alkylating agents. So basically, alkylating agents are a kind of agents which are generally useful for the purpose of alkylation because of their specific feature, what is called this dichloro, or we can call chloroethylamine functional group, chloroalkylamine functional group, which is present in such kinds of alkylating agents, in such kind of this so that they can do the cross linking of DNA. Generally, we know that DNA, what the model which was first identified by Watson and Crick. So this model is generally composed of this double helical structure. So this double helical structure of the DNA that is having so many hydrogen bonds in itself. But our these alkylating agents, majorly they are going to perform the task, or what is called this cross-linking of chains of DNA. So whatever the chain of the DNA through an alkylation, that will be cross-linked. So because of this, the processes, subsequent processes such as transcription and translation, so these are the processes shall be altered, and we can arrest the cell division, or we can prevent the multiplication, proliferation of the cells by cross-linking of strands of the DNA. Okay, so what are the agents used for this purpose? With chloramine, that is one of the agent. As we can see in its structure, this chloroethyl functional group is present, so that can basically use in an alkylation, and where nucleophilic nitrogen is there. So this is a CH2 which is electrophilic in nature because it is attached with a good living group, chloride. So chloride is considered as a good living group. So the nitrogen genus bases such as genus bases such as adenine, guanine, cytosine, and uracil, or majorly thymine. Uracil is not present in DNA. So these four of the nitrogenous bases, where they possess so many nitrogens. So one of the nitrogen can, any one of this nitrogen can attack on the CH2 carbon and can undergo an alkylation. Although I have not shown any kind of in alkylation processes in this slide. But if you visit this this medicine chemistry by Wilson and Giswold, you shall be able to see their mechanistic process where they have explained how an alkylation can be achieved. Okay, so methchloramine is one of the function this alkylating agent. Another alkylating agent is cyclophosphamide. Cyclophosphamide is basically a prodrug which is metabolized to aldophosphamide, and aldophosphamide that will perform this cross-linking of the DNA. So basically, it is a prodrug, cyclophosphamide, and it will form aldehyde. So that's why it is called as aldophosphamide. Then one more thing we would like to share with this drug. Cyclophosphamide is generally metabolized to aldophosphamide and another intermediate called as acrolein, having a carbon-carbon double bond as well as the aldehyde function. So this acrolein that will undergo it is toxic in nature because it can cause hemorrhagic cystitis, the vaginal bleeding. It can cause. So to prevent this side effect, N-acetylcysteine or glucosamine, they are given in combinations to prevent the occurrence of this hemorrhagic cystitis. Okay, so cyclophosphamide is that drug, and उसका ही एक isomer है, that is ifophosphamide. The isomer of cyclophosphamide is called as ifophosphamide. So this ifophosphamide, which is over here, basically the third alkylating agent which we are going to discuss, having similar mode of action, but the advantage is that we have it possesses basically less amount of side effect as hemorrhagic cystitis. Chlorambucil, that is a third alkylating agent, and in the structure we can see there is a butanoic acid which is present over here. Normal unbranched alkyl chain is present in its structure, as well as this dichlorone, but chloroethyl spacer is present on this nucleophilic nitrogen. Okay, and it can cause the cross-linking of the DNA. Bifalan, that is another alkylating agent based upon this fluorophenylalanine amino acid. Then we have seen some other classes, ethylamine, thiotepa. So basically, acidity in three-membered nucleus ring is present in its structure, and that possess uh, anti-cancer activity. Triethylenamine and busulfan. So busulfan is basically sulfate derivative, SO C minus derivative, and here basically triethylmelamine. Triethylmelamine is the uh, possess this 1,3,5 triazine benzotri, or we can call triazine as a nucleus in its structure. Nitrosoureas. So basically, these are the all nitrosoureas, which are derivatives of the urea. You can see this is carmustine, lomustine, and streptozotocin. So all of these basically nitrosoureas, they possess this something called urea functional group in its structure, as well as nitroso functional group. This NO is called as nitroso. So we can have, let us say, nitro is NO2. We can have NO3 minus, that is nitrate. We can have NO2, that is nitrite. And we can have NO, that is, sorry, NO2, NO3 minus is nitrate, NO2 is nitro, NO2 plus is nitronium ion, and NO simply that is called as nitrosonium ion. So this nitrosoureas basically carmustine and lomustine, and this another drug streptozotocin. You can see this is also cyclourea derivative. यहाँ पे six-membered pyran ring is present, which is basically sugar molecules in the structure of streptozotocin, and the streptozotocin, which is basically under clinically also used to induce diabetes in the experimental animals. Okay. So with this, on the class we have discussed that is triazine compounds. So procarbazine, dacarbazine, and one more drug that is temozolomide. They are triazine derivatives generally. Why they are called as triazine? As such, there is no triazine nucleus is present in their structure. But what happened that they possess majorly more three or more than three nitrogens. That's why they are termed as triazines. Platinum coordinate complex, cisplatin, carboplatin, oxaliplatin, these are the platinum compounds because cisplatin is totally inorganic molecule, which is what soluble one and having cis geometry in its structure. And this is basically cis bischloro bisammonium platinum 2 is the IUPAC name of cisplatin, and that possess the inhibitory activity for what is called uh, alkylating agents. So it can perform the cross alkylation of the DNA and can cause the inhibition of uh, or the can inhibit the tumor or can inhibit the proliferation of particular cells. Friends, now we are going to learn the new topic for today. Till the time we have discussed alkylating agents and their subtypes. Today is a day for discussion on some extraordinary classes or some additional classes for anti-neoplastic agents. And this agent is basically antimetabolites. So what are called metabolites and what are called antimetabolites? Whenever in a basically dictionary point of view, the meaning of anti is always to opposite. Just like we have dictionary wise, if we go, then if somebody is doing some, let us say, microorganisms. So we know that some microorganisms such as bacteria, virus, and fungus. So they basically can cause pathogenic conditions, pathogenic diseases. If we want to treat them, then whatever the agents are used, they are termed as antiviral, antibiotic, anti-fungal, anti what's diabetic. So anti is basically to counteract, to counter cancel the effects caused by the subjects. or the some other objects. So anti-metabolite, the meaning is anti basically means to opposite, to make the changes opposite. Metabolites means whatever the metabolites are there, so they are going to be the part of metabolism. Let us say you have already visited this carbohydrate metabolism, lipid metabolism, peptide metabolism in your first year Pharm D in your bioorganic chemistry. Okay, so what are called these bioorganic agents? Antimetabolites. So antimetabolites are the agents which are going to interact with the substances, the enzymes, the proteins, and they are going to inhibit. Sometimes they are also termed as fraud metabolites. Fraud is not a new term for you. Like just like it is a simple thing that a teacher is taking a class and I am taking the presence. So while marking the presence, sometimes let us say if I told roll number 21, so instead of so the roll number 21 is of Mr. or Mrs. XYZ, Mr. or Miss XYZ. So he or she should answer the roll call to mark his or her presence. But instead of roll number 21, if roll number 25 purposefully, wrongly, by doing some misbehavior, mark the presence at the place of 21, the roll number 25. So I'm not giving a specific example for that, but this is basically a kind of misbehavior. And because of what happened, 21, which was actually absent, now uh, he is or she is marked present. Okay, तो ऐसा हो सकता है. So anti-metabolites are also similarly these kinds of uh, behavior is there, basically fraud behavior. So microorganisms basically, or they possess, or this cellular environment, they possess, although they have good amount of affinity, agonist and antagonist ये जो define हुआ है ना, वो एंटा इसके बेस पर ही हुआ. लेकिन what can happen that sometimes this whatever the cycles which are occurring in metabolism, they can be made full with the help of these antimetabolites. So foolishness can be created in can be also do can be created in this. A kind of cheating can be done in metabolism as well. And that cheating basically here it is used as anti-neoplastic agents, or also called as fraud nucleotides. Or you will visit one of the substance 5-fluorouracil, which is metabolized to 5-fluorodeoxyuridine monophosphate, then diphosphate, triphosphate, का उसका further metabolism करके. But in fact, in that case, 5-fluorouracil is not required. We require only uracil monophosphate. उसके बदले 5-fluorouracil monophosphate है तो भी काम बन जाता है. So that's why it is called as fraudulent nucleotide. So we will visit that here house structure 5-fluorouracil. Okay, so it's not a big structure as well. Uracil is basically nitrogenous base, pyrimidine base, which is present in DNA. But instead of using uracil, if we use 5-fluorouracil, क्या होगा कि 5-fluorouracil will take part into the metabolism. It will metabolize to 5-fluorouridine monophosphate, diphosphate, then triphosphate. वो triphosphate आगे चल के फिर DNA में incorporate होना चाहिए. पर इस केस में वो नहीं होगा. सो नहीं होगा तो बिकॉज ऑफ दिस इट कैन अंडरगो wrong metabolism. इसलिए this is used as anti-cancer agents. So not only that, anti-cancer agents can be used for this purpose, but so many kinds of they are generally can be used. So and you might have heard that this is also a substance which is used for the purpose of, let us say, simply this antifungal agents, 5-fluorouracil and another substance that is amphotericin B. So they are given in combinations because amphotericin B will do the perforations in the fungal cell wall, and 5-fluorouracil can undergo there and can do the metabolism. Okay, so that was the case for this, and basically the side effects are that it can cause megaloblastic anemia. It can cause the anemia is a major side effect of this because it can cause this deficiency of vitamin B12, cyanocobalamin. That is the name of vitamin B12. Another substance after 5-fluorouracil, that is fluorofloxuridine. Well, again, it is based on the structure of very much similar to 5-fluorouracil, but the difference is that now it is a nucleoside. This was basically nitrogenous base, 5-fluorouracil, and it is attached with the sugar. So whenever a nitrogenous base is attached with the sugar, it is termed as nucleoside. Okay, and if nucleoside is attached with the phosphate, now it is called as nucleotide. Okay, so you should revise your knowledge on the nitrogenous bases. What are they? What are the structures? What is called nucleoside? What is called nucleotide? So nucleoside is basically a sugar nitrogenous basis attached with the sugar is called as nucleoside. So basically this is also nucleoside, floxuridine, and another drug is capecitabine. Capecitabine basically it is a prodrug of another ज़्यादा prodrug तो नहीं पर capecitabine and gemcitabine basically they are cytosine analog. Okay, so more clearly we can see the structure of cytosine. Only the difference is in uracil and cytosine is here we have a double bond O, but in case of cytosine we have NH2 amino functional group at this position. Okay, so capecitabine and gemcitabine basically they are cytosine analog, and cytarabine is also cytosine analog, just like 5-azacitidine, and this is also cytidine and cytosine analog. Nitrogenous base are of basically two types. What we can say purine type of the bases and pyrimidine type of the bases, where purine type of the bases where adenine and guanine, they are purine type of bases. Pyrimidine type of base such as cytosine and uracil and thymine. So they are monocyclic these bases. So here this cytosine analog, that is capecitabine, and tegaphor is also that kind of analog. But the structural difference in capecitabine is that here fluorouracil is connected with this 5-membered sugar, and sugar that is followed by here there is a presence of this ester n-butyl ester linkage in capecitabine. Agaphor, this is basically another heterocyclic nucleoside because the nitrogenous base attached with the sugar. The difference is that only one hydroxyl functional group is there. तो शुगर में नाम ही ऐसा है कि शुगर्स आर बेसिकली कार्बोहाइड्रेट्स एंड कार्बोहाइड्रेट्स आर पॉलीहाइड्रोक्सी एल्डिहाइड्स पॉलीहाइड्रोक्सी कीटोन्स. तो इसमें इस केस में तो ओनली वन हाइड्रोक्सी फंक्शनल ग्रुप है, और दूसरा भी एक होता है जो ग्लाइकोसिडिक लिंकेज जो ऑक्सीजन को लेके जो बना हुआ है वो प्रेजेंट नहीं होता, तो इट कैन बी हैविंग आइदर कीटो और एल्डिहाइड एज वेल. ठीक है, so here the difference is this n-butyl ester linkage is completely absent in this case because here now it is not this cytosine analog, but it is uracil analog, and uracil analog, since it is attached with uracil, so it is termed as uridine. This is basically uridine analog. If cytosine के साथ अटैच हुआ है तो उसको बोलते हैं साइटिडीन, यूरेसिल के साथ अटैच हुआ है तो उसको बोलते हैं यूरिडीन, और थाइमीन के साथ वो अटैच होता है तो उसको बोलते हैं थाइमिडीन. Then gemcitabine is also that kind of cytosine analog, but the structural difference is that in the sugar portion, the C2 position of the sugar is composed of two fluoro groups. Okay, and this gemcitabine and cytarabine basically they are going to act against DNA polymerase enzyme. Okay, so cytosine arabinoside analog, this gemcitabine, and this will going to inhibit. This is DNA polymerase enzyme as well. 5-azacitidine, 5-aza इसलिए बोला है क्योंकि फिफ्थ पोजीशन पे जहाँ पे फ्लोरोयूरेसिल का फ्लोरीन कार्बन अटैच कार्बन था, उसी जगह पे यहाँ पे 5 एजा ग्रुप रख That's why it is termed as 5-azacitidine. Okay. Then D-cytabine basically here the difference is dihydroxy and 5-azacitidine and D-azacitidine. Only difference is this hydroxy group C2-hydroxy C2 alcohol group which was present in the previous molecule called 5-azacitidine, that is now completely absent in D-cytabine. Yeah. So another drug is now we were up to pyrimidine analogs, basically 5-6 member ring and having the two nitrogens in its structure. Kabhi kabhi three bhi tha, that was called as SITD and D-cytidine. So that is basically this anti-cancer drug, pyridine analogs. And now we are going to pyrimidine analogs. Now we are going to have a discussion on another kind of antimetabolites, that is purine analogs. Purine are nothing. Purines are 5 membered bicyclic compounds having 4 nitrogens in its structure, and उसमें भी pyridine वाला जो रिंग इस साइड का होता है उसमें 2 नाइट्रोजन होता है, और इस साइड का जो 5 member रिंग है उसमें भी 2 नाइट्रोजन होता है. बट pyrimidine में 1,3 पोजीशन होता है, इसमें भी 1,3 होता है. और आप इसको pyrimidine बोल सकते हो. यहाँ पे इस साइड की जो रिंग है उसको imidazole we can call it. Okay, so this benzimidazole का सिंथेसिस आपने जैसे किया है ना वैसा ही दिखता है, बट डिफरेंस इज दैट benzimidazole में बेंजीनॉइड न्यूक्लियस होता है, हियर इट possess as a or nitrogen instead of carbon. So that's why it is termed as heterocyclic. Okay, so 6-mercaptopurine and thioguanine are there. So basically, this is 6-mercaptopurine, where the difference is that at purine का ही structure है. Purine आप ये SH हटा दो ना तो it is nothing, but it is called this mercaptopurine. And mercaptopurine का basically I am not sure whether you will visit the structure of one more anti-cancer drug that is called azathioprine. अच्छा नाम तो ऊपर दिया है पर नीचे उसका स्ट्रक्चर नहीं दिया है. ठीक है, मैं बोल देता हूँ आपको. दिस azathioprine is basically heterocyclic compound having nitrogen in structure. सल्फर भी होता है, प्यूरिन का स्कैफोल्ड है इसीलिए नाम बोला azathioprine. 6-5 और 6-azathioprine ऐसा कुछ होता है, I do not remember exactly, but this azathioprine is used as immunosuppressant. What is called immunosuppressant? For example, you are aware about organ transplantation. Okay, so organ transplantation is one kind of condition where the doctors will perform the transfer of the organs. जैसे आपने हेलो ब्रदर मूवी देखा होगा, तो what happens in that movie? What after the Salman Khan is shot and after his death, the heart of Salman Khan, उस फिल्म में उसका नाम हीरो था. तो जो हीरो का हार्ट है वो आगे चल के दूसरा उसी का ही भाई है, अरबाज खान शायद, आई एम नॉट रिमेम्बरिंग. ही वाज अ पुलिसमैन इन दैट मूवी. तो इन जो हीरो का मूवी था वो इसको लगा दिया गया है. सो व्हाट हैपेंस, दैट इज कॉल्ड बेसिकली हार्ट ट्रांसप्लांटेशन. किडनी ट्रांसप्लांटेशन भी पॉसिबल है, और मेनी काइंड्स ऑफ अदर ऑर्गन्स आर आल्सो ट्रांसप्लांटेड सक्सेसफुली. सो अब नाउ कमिंग टू द साइंस पोर्शन. मूवी भूल जाओ, अब मूवी की बात नहीं कर रहा हूँ. So when an organ transplantation is done, so our immune system or the immune system of the patient will perceive the external organ as the anti-organ. Means why body will not easily accept the external organ, and it will try to create the immune responses. These immune responses, which are totally based on the immune system of the body. मतलब इम्यूनिटी जितना स्ट्रांग होगा ना उतना ही वो रिस्पॉन्स स्ट्रॉन्गर होगा, और इम्यूनिटी जितना ही डाउन होगा वो इतना जल्दी से असर करेगा. So this is a difference for strong immune system and the weak immune system. तो इस केस में patient who has undergone organ transplantation, who survived the patient these immunosuppressants are given, क्योंकि उसका जो इम्यून रिस्पॉन्स जितना भी स्ट्रांग होगा ना उसको डाउन करो, ताकि जो दूसरे का ऑर्गन है, फॉरेन ऑर्गन, it can be fitted well into the system. So azathioprine is this kind of immunosuppressant that is given to the patient at the time of organ transplantation. And what is the metabolism way? Azathioprine का जो metabolite है that is 6-mercaptopurine. Or 6-mercaptopurine is really responsible for its immunosuppressant activity, anti-cancer activity, and this way. Okay, so that basically I wish to share with you. So this azathioprine and 6-mercaptopurine, they are also used as anti-cancer agents. Then one more kind of the class, that is thioguanine. So basically here that is thioguanine structure is present. उतना ही difference है यहाँ पे basically purine का में यहाँ पे NH2 होता है. Basically it is adenine and guanine. तो adenine के structure में यहाँ पे double bond O होता है. I think here it is not that. यहाँ पे double bond होता है, पर वो यहाँ इस केस में सल्फर है, इसलिए थायो नाम बोला है. और यहाँ पे NH2 तो होता ही है ग्वानिन में, इसलिए उसको बोला है थायोग्वानिन. 6-mercaptopurine, 6 amino, basically 6 thio, 2 amino, maybe 4, 6 mercaptopurine, yes, 1, 2, 3, 4, 5, and 6. So that way they are given the names. Fludarabine is basically that is also uh, I can see adenine, that is adenine analog. Okay, so fludarabine is basically this is also 5-membered cyclic sugar, and this is basically vidarabine. So fludarabine and vidarabine basically these are purine nucleotides having activity. And as I have rightly said that they belongs to this adenosine analog. ये भी adenosine analog है, because its structure is much simpler to adenosine. इसमें दोनों क्लेडाराबीन में क्लोरीन होता है, तो विडाराबीन में क्लोरीन को डिसअपियर कर दिया, बट C2 पोजीशन पे एक हाइड्रोजन को. ऑल्दो यू विल, ये जो यहाँ पे जो बॉन्ड लिखा है ना, उसका भी एक स्पेसिफिक कॉन्फ़िगरेशन होता है, वेदर इट इज एक्सियल और वेदर इट इज इक्वेटोरियल. ये एक्सियल और इक्वेटोरियल जो नॉमिनेशंस है, काइंड ऑफ स्ट्रक्चरल कंपोजिशंस व्हिच यू पीपल माइट हैव विजिटेड ड्यूरिंग योर साइक्लोहेक्सेन केमिस्ट्री, कि कौन सा बॉन्ड एक्सियल है, कौन सा बॉन्ड इक्वेटोरियल है. सो एट दैट टाइम यू माइट दिस. One more drug, that is pentostatin and fludarabine. So pentostatin may be 6 membered nahi hai, but this time it is basically 7 6 5 membered ring, and fludarabine basically 6 5 membered, because here it possesses fluorine, again adenosine analog hai, and this is a sugar, but sugar possesses one phosphate functional group. So fludarabine is basically a kind of nucleotide. Okay, so this way it possesses this. These are all the structures of fludarabine, pentostatin, azathioprine, and this. Common suggestion to all of you that in medicinal chemistry, basically it is difficult to remember the names of the these structures of these drugs. What we can do, we can generally recognize their names. जैसे कि आप नाम याद रखो, थायोग्वानिन, so it possesses guanine nucleus. Then vidarabine, तो it possesses adenosine nucleus, because उसके नाम में कहीं ना कहीं आपको adenosine का ए का pronunciation देखने को मिलेगा. You can remember likewise. So इसी तरीके से you can make your shortcut keys to decode the name into the cyclic structure. बहुत ही if you could not remember the properly the whole structure of a drug, what is connected with what, if you could not remember that, simply you can go with the remembering the heterocyclic ring which is present. Folic acid analog, this is a one more kind of the analog, basically folic acid analogs, methotrexate and pemetrexate, basically they are drug. So I shall show you the structure of this folic acid. So this is folic acid structure. We can divide the structure of folic acid into three equal parts. So what are these parts? The part number one, that is here, that is pteridine ring. This is the ring which is present over here, 6 member bicyclic compound having 4 nitrogens. So that is termed as pteridine ring, and this pteridine ring is basically uh, one of the important component, right? So pteridine ring is present in folic acid. उसके बाद this is nothing but this is para amino benzoic acid, PABA. So folic acid के structure में para amino benzoic acid is present, and that PABA is conjugated with this. I want to say this one, glutamic acid. So glutamic acid is the major component of folic acid. So folic acid के structure को you can divide in three equal parts and can remember for that one: pteridine ring, para-aminobenzoic acid, and this another component what is called as this one, uh, glutamic acid. Okay, so now we wish to see what is the difference between the folic. You know, for folic acid is nothing but that is vitamin B9, which is majorly present in green leafy vegetables. So at this age, you might have observed that many of us or many of your parents, many of your let us say your family members may have the deficiency of vitamin B9, folic acid. So what they are taking after, they might be taking the medicine, multivitamin tablets, and that majorly composed of folic acid. But if you have a habit of eating green leafy vegetables, such as your salad, जो आप टोमेटो, कैबेज, पालक और कैरेट और जो भी सब खाते हो, so that is a potential source of folic acid, and basically we can utilize that folic acid directly from our food. एक आपने जब एंटीबैक्टीरियल और एंटी सल्फोनामाइड का आपका जब चैप्टर हमने पढ़ाया था, तो उस टाइम पे वी हैव डिस्कस दैट bacteria cannot utilize this kind of folic acid directly. It's only we can utilize that folic acid. We, the human beings, we, the Homo sapiens, can utilize that folic acid directly. Okay, directly means from the food we can utilize, but bacteria will not utilize the folic acid from the food. They will be dependent upon the folic acid which is present in our body. Okay, so that is the major difference. So folic acid or methotrexate, basically methotrexate is folic acid antagonist, which where will be this folic acid is essential for one carbon metabolism. क्योंकि वो जो यूरिडीन से मोनोफॉस्फेट से ये बनाता है थाइमिडीन मोनोफॉस्फेट. So basically DNA requires thymidine, not the uridine. So that one carbon metabolism, one C metabolism is essential with the help of this folic acid. Okay, so here this methotrexate, this उतना ही difference है, the same structure of folic acid, but आपका जो OH था वहां पे आपने NH2 बना दिया, और जहां पे आपका folic acid में NH था वहां पर आपने N-methyl कर दिया. So now it is completely different, folic acid and methotrexate. So one more drug in this category, that is pemetrexed and trimethoprim. So, but difference is lot of structural difference is that here it was a 6 member ring, but now it is converted into 6 member ring. Not only that, आप folic acid में जो OH था उसका आप ketone. That is one more difference in the structure of folic acid versus pemetrexed. Then another drug is trimethoxate. So trimethoxate में पूरा ग्लूटामिक एसिड को हटा दिया है, फिनाइल रिंग को रखा है, पर फिनाइल रिंग में सब्स्टिट्यूशन कर दिया है 3 methoxy group. 3 methoxy groups are present on third, fourth, and fifth position. So trimethoxy benzene ring is present in such folic acid. Okay, and iska basically they are folic acid antagonist. Folic acid is essential in one carbon metabolism, and if we have deficiency of folic acid, so it will result in formation of anemia. Okay, and these folic acid antagonists का side effect भी वही है, they are going to cause anemia. Anemia is basically a disorder of malformation of hemoglobin, or some there may be less formation of hemoglobin, so that is called as anemic conditions, anemia. And there are so many types of anemia: pernicious anemia, megaloblastic anemia, normochromic, normocytic, hemochromic, hypohemochromic, depending upon the level of hemoglobin. Also different types of anemia can be there. So now we are coming to the last, not the last one, but basically one of the main important after the alkylating agents and after this end of this platinum compounds. And third class we have that all also seen basically this antibiotic antimetabolites. Now we are going to visit one more class, that is basically anti-cancer antibiotics. So what are the antibiotics? Antibiotics are nothing but simply we can call them as substance which are generally causing the inhibition of microorganism. But the advantage is that their potential source is generally called microorganisms. So इसीलिए antibacterial agent और antibiotic में different नहीं है. Antibiotics are generally obtained from microorganisms, from fungus cells, from virus cells, etc., etc. And this uh, and other substances such as bleomycin, mitomycin C, anthracyclines, etc. They are the kinds of these antibiotics, and they are obtained from microorganisms such as this actinomycin D, which is obtained from soil bacterium. तो जमीन में जो माइक्रोबैक्टीरियम जमीन में actinomycin D. Okay, and mitomycin, and similarly we can identify their specific sources. So this antibiotics, which was first discovered by the scientist Sir Alexander Fleming. And you know what is the antibiotic? Penicillin, which was obtained from Penicillium notatum, Penicillium chrysogenum. So he was the first chemist to identify this penicillin antibiotic from this microorganisms, and that was identified during the time of World War. Okay, so बाद में Fleury and Cheng were the another two scientists who have utilized this multi-scale synthesis of the same antibiotic in their labs, and this was basically a record break, a kind of breakthrough drug in the field of drug discovery, because at the time of World War, this penicillin was more required. फिर वो जो सैनिक होते थे वो वॉर में जाते थे, लड़ाई करते थे, फिर उनको इंजरी होती थी. To prevent themselves of injury, to recover on the next day, to fight with the enemies, they require this antibiotic. इसीलिए many a times defense of any country also promote the research. Defense research the organization, DRDO, Defense Research Organization. So that is one of the organization in our country, DRDO, which promotes the research, and that is on these kinds of agents, antibiotics, sometimes maybe some other agents. So that basically promotes the research of new compounds. उसमें funding they are providing. So for the purpose of this, that how one can recover from these kinds of injuries. Okay. So now coming to our another this structure of antibiotics. So these antibiotics are so many kinds of this. Actinomycin D is also called as actinomycin, and that is like possessing its structure is possess the cyclic peptides, as well as यहाँ पर भी एक ring है उसको बोला phenoxyl ring, so that is present in the structure of actinomycin D or bactinomycin. Both are same, obtained from soil bacteria. I do not remember the name of that soil bacterium, but if you visit medicinal chemistry by Wilson and Griswold, you will find that recorded name on the pages of this uh, book. Mitomycin C, that is also another antibiotic, and here it possesses this pyrrolidine nucleus as well as aziridine ring structure. Doxorubicin, down basically, they are also this kind of anti-cancer agents. So they also possess this kind of substance where it can show that this antibiotic activity. Idarubicin and abirubicin, they are also these kinds of antibiotics. And the one more class we have to see, that is the final, the last class for today, that is plant products. So they are also more important in this regard. Okay, so we shall cover these slides, or the last two slides for the sessions. Plant products. Natural products are excellent component for cancer, and I mean for the therapeutic treatment of the cancer. રાઈટ તમે ઘરે હશો અને ત્યાં ડેકોરેટિંગ પ્લાન્ટ બારમાસી છોડનું નામ તમે સાંભળેલું હશે. Nothing but a kind of pink colored flower are there. So that is nothing but it is called as Vinca rosea, biological name of this, and family is Apocynaceae. So this is a powerful medicine for treatment of cancer. And the leaves and these flowers, basically leaves में ही ज़्यादा content होता है. They are potential source of vincristine and vinblastine. Okay, and this vincristine and vinblastine alkaloids, they are potent anti-cancer agents because the mother nature itself is made up of full of medicines. We do not have to many a times depend upon the synthetic medicinal chemical constituents. Just like you remember that turmeric, which is obtained from Curcuma longa, which is Gujarati માં જેને આપણે કહીએ તો હળદર. That is a powerful medicine to treat yourself from COVID-19. You can call. It is basically immunomodulator. It can help to improvise your health. થોડી પણ જરા શરદી ઉધરસ કે તાવ કે એવું આવે ને હળદર વાળું પાણી પી લો, હળદર મીઠું કરીને ઉકાળો કરીને પી લો, એટલે જુઓ બીજા દિવસે ઇમ્યુનિટી સ્ટ્રોંગ હશે. That is possible through the plant products. It's possible through the natural products, the phytochemicals. That's why I also will also suggest to you people to make the use of medicinal make the use of food of medicinal importance in your daily routine. That is more important. એમાં એવું ના હોવું જોઈએ કે કરેલાનું શાક છે કડવું લાગે છે એટલે હું ખાતો નથી કે ખાતી નથી. No, you should focus rather on the taste. बिना हड्डी की जो जीभ है उसके अलावा, rather than following the taste, you should focus on the merit of your food item. तभी आप you will be able to survive for longer period of time free from any disease conditions. Otherwise फिर B9 की डेफिशिएंसी है, हीमोग्लोबिन की डेफिशिएंसी है, B12 की डेफिशिएंसी है, whatever can occur if you do not follow this. If you do not involve the food of medicinal importance in your daily routine. तो जो daily food stuff है उसमें ये सारी चीजें होनी चाहिए. Then these things can be more important. And that is actually this slide is telling. Vincristine, vinblastine, basically they are this kind of anti-cancer agents. Okay, and they are used. Podophylletoxin, etoposide, and paclitaxel. And Taxol is one of the blockbuster anti-cancer drug, which is obtained from Taxus baccata and Taxus brevifolia. Family is Taxaceae. This was one of the blockbuster anti-cancer drug molecule once upon a time. Okay, and that is a powerful medicine to treat the cancer. Paclitaxel. And look at the structure. The structure is highly complex. Paclitaxel भी है, paclitaxel भी आ गया है, taxon, taxanes भी, taxol भी है. Taxol का structure मैंने यहाँ पे नहीं दिया है, इस slide पे नहीं दिखाया है. But yes, paclitaxel is also this drug. And look at to see the structure of this drug, much complicated. It is not easy to synthesize this compound in one step. It requires more than 10, 15, 20, 25 steps. Initially, when the people have reported, तो उसके time पे atom economy, overall yield, and number of steps for the synthesis of this compound was there. Different people have given different approach. I do not remember their names. पर जैसे किसी का ऑर्गेनिक केमिस्ट्री लैब में कोई 25 स्टेप में पैक्लिटैक्सेल या फिर टैक्सोल बना लेता है, उसके लिए asymmetric synthesis का knowledge is required. It is not easy. Asymmetric synthesis is the synthesis which deals with stereochemistry. तो कोई 26 स्टेप में अपना टैक्सोल बना लेता था, तो कोई फिर 22 स्टेप में, तो उसके लिए वो तो दैट वाज अ न्यू डिस्कवरी कि उनके मेथोडोलॉजी में वो 26 step by step reaction से वो taxol बनाते हैं. And somebody has reported some new method where within 20, 16, 15 steps taxol can be synthesized. What the people have done. But for this you will require the knowledge of asymmetric synthesis, because look at the structure of paclitaxel over here. It is having so many chiral centers. These all bonds represents the chirality within the structure. Here also there are the chiralities, and this is somehow kind of cis configuration, E or Z configuration is required. Okay, so नेचुरल प्लांट में तो बनता ही है, बट क्या है, कितने आप नेचुरल प्लांट आप फिर कैंसर किया है, सो मेनी नंबर ऑफ मिलियंस ऑफ पीपल्स आर हैविंग कैंसर, तो उनके लिए दवाई बनाने में हम सारे झाड़ पान क्या काटते क्या? So alternative is to synthesize such kind of chemical constituents in the lab, and where organic chemistry can help us. Okay, so we can synthesize the compound using this. Podophylotoxin, basically podophyllum, etoposide, which is obtained from Himalayan yew tree. आप हिमालय में, वो नॉर्थ इंडिया में जब आप घूमने जाते हो तब वहां पर हिमालयन यू ट्री आपको देखने को मिलेगा, हिमालयन मे एप्पल, पोडोफाइलम. सो ये जो पोडोफाइलम इज कॉल्ड हिमालयन मे एप्पल, क्योंकि वो मे मंथ में, इन द मंथ ऑफ, दिस इज फेब्रुआरी, आफ्टर मार्च, अप्रैल एंड मे, द फ्रूट विल कम on this tree. That's why it is termed as Himalayan May apple, the name of the tree. And that from its bark, this chemical constituent is obtained, called as podophylotoxin. That is also one of the important chemical constituent. And the structural component present in it is benz 1,3-dioxol. So that is the chemical constituent. And cyclic lactone is also present along with this trimethoxyphenyl ring. And these are some other components. Camphothecin, which is basically obtained from Camphotheca, and I forgot Camphothecin. Camphotheca is the genus याद है, पूरा नाम मुझे याद नहीं. And that possesses topotecan, which is basically anti-cancer drug. Then some other vincristine, vinblastine के भी structure यहाँ पे है. Look at their structure. They are highly complicated. Vindoline subunit is present. Basically, they are indolene alkaloids, because in that indole alkaloids, because in that structure, indole ring is present. 6,5-membered bicyclic compound indole. You will be performing one synthesis in your lab using Fischer indole synthesis. That is for the synthesis of 6,5-membered bicyclic compound indole. So that indole is present in these two alkaloids. मुझे मैंने एक स्टेटिस्टिक्स ऐसा पढ़ा था जिसमें लिखा था कि आप कई गुना टन्स एंड मिलियंस ऑफ आप विनका रोजिया प्लांट को एक्सट्रैक्ट करते हो तब आपको हार्डली एक पेशेंट या 10 पेशेंट के लिए मेडिसिन उपलब्ध होगी इफ यू आर कटिंग tons of vinca rosea from their leaves or flowers or whatever chemical constituent बहुत minute quantity में you will be able to extract. So there are millions of patients of cancer. So how to prevent, how to give this medicine? Ideal way is to synthesize this compound in the lab. So that's why this synthetic organic chemistry is more popular for the synthesis of this fine phytochemicals. On the branch which is popular for synthesis of this final component that is called as total synthesis. Okay. So total synthesis of taxol, total synthesis of camphothecin, total synthesis of vincristine, vinblastine, they are popular. And organic chemist can synthesize all of these compounds. With this, we are going to stop up to this. In the next coming sessions, we shall discuss from here onwards. So thank you everyone for attending the session.